This is Dr. Vishnu Shankar Sinha, PG Department of Botany, Tata College, Chaivasa. Today I am going to discuss apoptosis or programmed cell death. In multicellular organisms, development and homeostasis depends on cell division, cell differentiation, and cell death. There are two methods that a cell can die, that is apoptosis and necrosis. Apoptosis is a Greek word, it means falling off, and apoptosis consists of two words, that is apo means from and tosis means falling. The term apoptosis was first introduced by Carretel in 1972. Apoptosis or programmed cell death is a very orderly, genetically controlled mechanism of cell death or cellular suicide of unwanted cells accompanied by elimination. Apoptosis is vital for embryological development and neurological pruning. This picture clearly shows that how a normal cell can die. It dies by two methods, that is necrosis and apoptosis. I have already discussed. In necrosis, the cell is injured by the external factors, while in apoptosis, the cell may be cell suicide by the internal or external factors. What are the biochemical and morphological events of apoptosis cell death? Number 1, shrinking of cell and nucleus, loss of potential to adhesion with neighboring cells, cell membrane form bubble-like blebs on the outer surface, occurrence of nuclear membrane break, dilation of endoplasmic reticulum and breakdown of mitochondria with release of cytochrome C, chromatin condensation and chromosomal DNA fragmentation, rapid engulfment of the apoptotic cell divides by nearby cells by the process of phagocytosis that clean up the cell fragments. Yes, this picture clearly showed the stages of apoptosis. Initially, in normal cell, the cell membrane and the nucleus get shrinking, then chromatin condensation. After that, membrane blooming and nuclear collapse. The formation of apoptotic body, lysis of apoptotic body. The apoptotic body is engulfed by the process of phagocytosis by the cells. Yes, the decision to perform apoptosis comes from the cell itself, from the surrounding cells, tissues, and maintain the regulation of tissue homeostasis. Recent reports have indicated that due to apoptosis, an average adult human loses 50 to 70 billion cells per day, and an average human child between the age of 8 to 14 years approximately 20 to 30 billion cells suicide per day. Apoptosis inhibits tumor formation, that is cancer or autoimmune diseases, while excess apoptosis may lead to neurodegenerative diseases and immunodeficiency. Necrosis. It is occur due to damage by external forces such as poison and infection and bodily injury or getting cut off from the blood supply, which may occur a heart attack or a stroke. Objective of elimination or kill of unwanted cells. Number 1, the cells have finished their own function, or cell having no function, cells that are produced in excess, cells that developed improperly, cell that is harmful or potentially dangerous for the survival of the organism, for development of homeostasis, or for defense and for aging, that is sensation. This picture clearly show that the extrinsic factor and intrinsic factor regulate the process of apoptosis. The extrinsic factors are cytotoxic drugs, nutrient loss, radiation, ultraviolet lights, hormones, heat, X-ray, etc. These extrinsic factors impact on the BCL2 protein, and the BCL2 protein is produced from the nuclear DNA by the process of translation. These BCL2 protein in association with the extrinsic factors, their decision has been taken and the mitochondria break down with secretion of the cytochrome C. And intrinsic factors are signaling, manufacturing, malformed proteins, etc. Genes for apoptosis. The MYC gene drive both cell proliferation and programmed cell death. In the presence of MYC protein encoded by MYC gene, it has been earlier reported that gene P53 is responsible for apoptosis. Inhibitors of apoptosis. Gene like BCL2, which lies on chromosome number 18, and BCR-ABL or VABL inhibit apoptosis. It induced by agents like radiation, serum deprivation, and various toxic compounds. Other compounds which inhibit apoptosis are orine tricarboxylic acid, that is AZT, zinc ions, antioxidants, and cytokinins, etc. This picture clearly showed the gene of cell cycle involved in the apoptosis process or apoptotic regulatory genes, and the checkpoint activation and delay progression during the apoptosis. In the steps of the cell cycle, that is G1, S, G2 and M phase, how the genes impart or take part in the process of apoptosis. Cancer and apoptosis. Apoptosis mechanism quickly destroy any abnormal or damaged or harmful cells accompanied by elimination from body. If apoptosis does not occur, these damaged and abnormal cells may survive and develop into tumor, that is a formation of cancer cell. Apoptosis inhibit development of cancerous cell. Enzyme and apoptosis. The initiation phase of apoptosis is beginning by extracellular or intracellular stress, that is low oxygen levels or DNA damage. It is mediated by proteolytic enzyme called caspase that causes DNA damage, rearrange the cytoskeleton and disrupt intracellular transport, cell division and signal transduction. Apoptosis can start by perforin protein secreted by cytotoxic T cell. It is an immune cell. Perforin creates hole in plasma membrane of the target cell. After entering in the cell, they release granzyme A and B, that is a proteolytic enzyme that initiate the performance pathway and smash on cell structure and cell function. Thank you for watching. For detail, you may consult the Harper of Biochemistry and Molecular Biology of the other books. Thank you.